Hello, I will be discussing antigen presentation and lymphocyte maturation. Antigens are any molecule that result in an immune response. They can be made of polysaccharides, lipids, nucleic acids, or proteins, which elicit the strongest immune response. Bacteria, viruses, and the body's own cells are some antigens that can be recognized by the immune system. Antigens contain a structure called the epitope, which serves as a region for recognition by lymphocytes. Lymphocytes contain receptors on their surface called paratopes that serve as a binding site for the antigen epitopes. Dendritic cells are normally found in the body, tissues, and lymphoid organs. After phagocytosis, an extracellular pathogen, dendritic cells process antigen protein fragments for presentation through MHC class II on their cell surface. Dendrites displaying foreign proteins through MHC2 then travel to lymph nodes to activate T lymphocytes. Dendrites displaying foreign proteins through MHC2 then travel to lymph nodes where they activate naive T lymphocytes by stimulating CD4 and CD8 receptors. This encourages naive T cells to mature and differentiate into T helper cells and cytotoxic T cells. Dendrites may also present foreign proteins through MHC1, since this class is present on all nucleated cells. This leads to the activation and differentiation of naive T cells into cytotoxic T cells. This process of cross-presentation is important in antiviral and anti-carcinogenic protection. B cells are able to detect extracellular antigens without the help of display proteins like MHC. They use B cell receptors on their cell surface to interact with antigens and secrete antibodies that have the ability to neutralize antigens or tag them for phagocytosis. B cells undergo maturation in the bone marrow. Here they undergo various stages of selection to ensure that they are both functional and self-tolerant. First, they undergo positive selection. The resulting cells that have the ability to bind to antigens then undergo negative selection to prevent self-reactivity. Cells that are unable to bind to antigens or are self-reactive are tagged for apoptosis. After successful maturation, B cells then are able to circulate peripheral immune organs. Unlike B cells, T cells originate in the bone marrow but mature in the thymus. Here they go through positive selection to ensure the ability to recognize MHC molecules. Negative selection then ensures that these cells are non-reactive to self-antigens. Any T lymphocytes that are unable to bind properly and recognize MHC or are self-reactive undergo apoptosis. Mature cells can then participate in immune responses. The structure of secreted antibodies and B cell receptors are similar besides the fact that BCRs are anchored at the membrane of B cells. They are composed of two heavy chains and two light chains, each one containing one variable region. The paratope is found at the end of the variable regions to bind with epitopes of an antigen. There are approximately 30 to 45 segments in the variable region of each heavy and light chain of BCRs and on the alpha and beta chains of TCRs. Diversity of these variable regions is due to genetic recombination and allows for the generation of unique receptors for a wide range of antigens. This concludes the presentation.